Hello folks, this is Dr. Jain. I'm talking about the next few slides for about 30 minutes or so, the neurobiological origin of depression. This is part of the psychopharmacology lecture series that we have been undertaking as a part of your psychiatry clerkship third year rotation. So things that we'll be covering in this lecture, the objectives are, we'll be talking about the neurobiological origins of mood disorders. We're going to be talking about the main hypothesis that underscores the origins of the mood disorder, that is the monoamine hypothesis of mood disorders. Then we'll be talking about psychopharmacology of the antidepressants and the neurotransmitters. And the main neurotransmitters we'll be talking about will be the role of serotonergic, norepinephrine, and dopaminergic neurons in the influence of mood disorders. Let's take a few minutes to assess what the course overview looks like. We'll be first conducting a pre-test assessment. So before we go any further, I would like you guys to click on the link on the right hand side that will lead you to a pre-assessment self-assessment before we go any further. That will give us an understanding what is our level of understanding at present, and then we'll review the presentation and then conduct a post-presentation self-assessment, which will give you a good idea of how much learning you have done. Those questions which are enumerated are very much reflect the NBME style questionnaire, which is a combination of clinical assessment based on your basic science skills. After the presentation, we'll be dividing dividing our group into small formal group discussion sessions where a topic of your choice, again you can see the link on the right hand side, where clinical scenarios are being enumerated, which I'll be requiring you guys to work and discuss among yourselves and come up with one of you guys presenting the outcome of it. Finally, I'll be asking you to get some feedback from me in terms of how your post-test performance has been, in terms of how much you have learned based on the presentation. And at the end, you guys as my students will give me anonymous feedback about the presentation so that I can make some improvement and changes as necessary based on your feedback. So let's start, shall we? The first slide, again, all these slides are based on Stahl's Essential Clinical Psychopharmacology, which is an online version. A hard copy of this book is available in our department library and also in the school library. You can have access to these courses, these slides, again, online through the library web page. If you're interested, please see me after the lecture. I'll be happy to direct you towards the resources. So let's talk about the monoamine hypothesis of depression. There are three key monoamines: the dopamine, the norepinephrine, and the serotonin. You guys know this. This is a schematic way of showing there are three major pathways in terms of connecting the prefrontal cortex with the other areas of the brain, mainly the red representing norepinephrine, the yellow the serotonin, and the blue-green is with the dopaminergic. And again, when you see that when there is ample neurotransmitter transmission in these three neurons, there is no depression. Whenever there is deficiency of monoamines, as noted in the slide B, symptoms of depressions can originate. However, depression, as we know, is not just on neurotransmitters, it's an interplay of genetics and other environmental factors. But this is a very good schematic way of looking at the origins of depression through the monoamine hypothesis. And the next slide shows the monoamine hypothesis of antidepressant action. As you can see here, in this scenario, you find that the neurotransmitters being generated over here. Of course, you have the transmission of neurotransmitters through the synaptic cleft, and then you have a postsynaptic neuron over here. And then antidepressants, if this is an antidepressant you've been giving, it's a reuptake inhibitor. Basically, they stop the reuptake. They stop the reuptake of the monoamines. They stop this action, causing more of the neurotransmitter in the synaptic cleft over here. And basically, one way or the other, they cause more hitting, the more neurotransmitter hitting the post synaptic neurons, leading to the effect of the more transmission is there, the more availability of neurotransmitters there in the neurotransmitter cleft, the better the mood symptoms can be. In depression, of course, the classical monoamine hypothesis depression suggests that decreased activity of the neurotransmitters. Again, we can go over here, indicating that. Let me bring up my tool over here. And again, this one again shows this area. It shows the wrong tool. This area again is the area, for example, of prefrontal cortex, showing deficiency of neurotransmitters, leading to paucity of neurotransmitter signals going into the other parts of the brain, leading to origins of depression. So basically what it's saying is that when there is paucity of neurotransmitter, there is upregulation. This upregulation of postsynaptic neuroamine receptors, leading to depression. This can be regarded as one of the markers. So upregulation is the key concept over here. Upregulation happens when there's paucity of neurotransmitters, and that's one of the pathognomic sign of development of depression. Another slide that shows how the depression works. So again, this slide shows the action of antidepressants. So in a depressed state, for example, if you see this upregulation of neuroreceptors at the postsynaptic neurons, addition of a for example, addition of or availability of a neurotransmitter through way of a antidepressant will cause more hitting of the receptors by the neurochemicals, leading eventually in the slide B of downregulation. That regulation means that as more neurotransmitters are available, less receptors will be available within due course of time, and this downregulation of these receptors can be correlated with the onset of action of antidepressants. And these receptors take time to be downregulated, and that one of the reasons we explained that it takes such a long period of time, about 2 to 3 weeks at least, for the clinical onset of action of neurons. Next slide. Again, we see, based on the monoamine hypothesis, antidepressants can theoretically create a return to normal mood state by increasing the level of neurotransmitters. As you can see here, antidepressants, when added to it, they cause medications through number of cascades of reactions, can cause action to the DNAs, to certain receptors, causing upregulation of proteins, leading to increased synthesis of various proteins. One of the increased proteins that is closely correlated with onset of antidepressant effective medication is the BDNF. This is a brain-derived neurotrophic factor, and which leads to neurogenesis and ultimately response to the treatment. The time course of the antidepressants, of course, receptor sensitivity is there. Again, the more down regulation of the effect happens, as more and more receptors are down regulated, the more antidepressant or the clinical effect of antidepressant becomes effective. So the course of action, as I said before, it takes time to take action as the neuroreceptors are being down regulated at the same time the clinical effect takes into effect. And this may translate into over weeks. Ideally they say up to 6 to 8 weeks, but there have been studies that up to 12 weeks, it may take up to 12 weeks to have full therapeutic benefit of antidepressants. The next slide shows about the synthesis and the metabolism of serotonin. As we see over here, serotonin is one of the major neurotransmitter that is responsible for regulation of mood. Again, serotonin is known by 5-hydroxytryptamine, 5-HT, as you all know. Its precursor is synthesized from monoamine tryptophan, and then it goes through a number of different cascades of reaction, tryptophan transporter being one of the rate limiting step that takes in tryptophan. That's the neurotransmitter transporter, tryptophan A in slide A, and then is pumped into serotonin neurons, and it is hydroxylated at the rate limiting step. For 5-HT synthesis, It's called tryptophan hydroxylase. Again, synthesis is a happens in presynaptic neurons, and it is stored in the vesicles, and ultimately it is secreted in the synaptic cleft. Now, the on the slide B, you see the serotonergic transporter that is responsible for reuptake of that. So, 5-HT released into synapse during transmission is either transported back, that this way it is being transported back to it, or it can be destroyed by MAOI-A or MAOI-B. These are two major mechanisms. Other things they can dissipate through the bloodstream outside. So there are three ways actually that the serotonin is dissipated. As you would know that stopping of this mechanism, which is very powerful by antidepressant, leads to more availability of serotonin in the synaptic cleft, hence the name serotonin receptor uptake inhibitors. This is a much more details of that. One of the transmitter profile that we see is presynaptic 5-HT1BD. As you can see over here, these are two major neurotransmitters that have got significant role in transmission of neurotransmitter transmissions. Now, 5-HT1B and D receptors are located in the axon terminal A. You can see the axon terminal here, and you see they are here also. These receptors further inhibit 5-HT release when they detect the presence of 5-HT in the synapse. So this is one of the ways that ways where it prevents excessive intake or excessive depletion of neurotransmitter in the synaptic cleft. Again, there is another receptor, 5-HT1A receptors, located in the neuron, which is in the axon body, which is also has got significant say on how much receptor is being, how much neurotransmitter is being released. For example, 5-HT1A is a somatodendric autoreceptor. Stimulation of 5-HT1A receptor leads to paucity. It kind of decreases, and we will see the clinical efficacy, how does really translate clinically into that. So if you see 5-HT1A receptor agonist, you probably are noting that it's causing less release of serotonin in the synaptic cleft. Serotonin receptor, this is the part 3 of the receptors. Now, this is where clinically they define what each receptor is clinically responsible for. For example, 5-HT1A. Now, stimulation of 5-HT1A has got some effect on the hormones. It definitely got effect on depression. It's got effect on anxiety, cognition, and inhibition of cortical pyramidal neurons. So these are the effects which are known to be associated with 5-HT1A. 5-HT2A, for example, is associated with sleep. It can be associated with hallucinations, inhibitions of dopamine release. It can cause excitation of glutamate release, which is an inhibitory neuron, and cause excitation of pyramidal neurons. The 5-HT2C, of course, is responsible for obesity, mood regulation, cognition, and regulation of dopamine and norepinephrine. This is significantly important, and you will see when we correlate this clinically with the antidepressants, you will see a strong correlation there. And there are other minor 5-HT3 and 5-HT6 and 5-HT7 which are responsible for a minor difference. Like, for example, 5-HT6 is responsible for long-term memory, and 5-HT3 involved in vomiting and regulation of inhibitory neurotransmitters, and 5-HT7, for example, this question is sometimes asked, is regulated in sleep and circadian rhythms. So that's something you may be asked questions in terms of what receptors are there. This is another slide which is, I'm going to enlarge this a little bit so you can see it. Similar properties as we just mentioned there. And this is another slide where there are, this is the projection that we see in the serotonin projection. So you can see the serotonin projection not only connecting the cortical areas, but it connects a number of other parts of the brains, including the striatum, the prefrontal cortex, um, the thalamus, um, the nucleus accumbens, the hippocampus. So hence because serotonin pathways are connected to so many different important areas of brain, so it is not surprising that serotonin has been implicated in control of mood, appetite, suicidal ideations, and sleep. This is just a schematic way of showing that how far it is connected to the various part of the brain. Now we move on to the next major neurotransmitter synthesis and metabolism now of norepinephrine. So as I was mentioning, this is the major synthesis and metabolism of a norepinephrine I was talking about. Norepinephrine is the rate limiting enzyme is, of course, as we know, from your basic science courses, is of course the tyrosine transporter. That's the main rate limiting thing, and then you have uptake of tyrosine, leading to formation of ultimately formation hydroxylated and transported to dopa, uh, of course, by the rate limiting enzyme for the non-epinephrine synthesis, namely tyrosine hydroxylase. That's the main rate limiting enzyme. And then dopa is finally decarboxylated to dopamine by the enzyme dopamine or dopa decarboxylase, DDC for short. As you can see, DDC has been, and ultimately you see in the neuron synthesis stops over here. However, a non-epinephrine neuron, and this is where the dopamine stops, and in non-epinephrine, however, dopamine is hydroxylated to non-epinephrine, so it continues to them the cascade of neurons. This probably you are familiar with in your basic science. Now once non-epinephrine is stored in the synaptic cleft, is transported inside the vesicle via VMAT2 or vesicular monoamine transporter type 2, also known as synaptic vesicle pump for monoamines. And once released, it can be, of course, it can go through reuptake, as we've seen in serotonin. It's non-epinephrine transporter reuptake. We also see that it can be metabolized by COMT, which destroys non-epinephrine, and it destroys through MAO-A and MAO-B. So that is the life cycle of non-epinephrine synthesis and metabolism. Let's go to the receptor profile again. Again, you see here that norepinephrine has got a number of different receptor profile. Here again, you see the non-epinephrine transporter net, which is responsible for reuptake of neurotransmitters from the synaptic cleft, and you see a number of receptors, including alpha-1 receptors, postsynaptic alpha-2 receptors, alpha-2b, alpha-2c, beta 1, beta 2, beta 3, and the number of there. And then you have presynaptic alpha 2 autoreceptors. And we're going to talk about also some of the key distinguishing features we have for each of the alpha receptors in a second. So next slide, of course, shows the this slide again continues to emphasize on the role of alpha 2 receptors, which are heavily responsible for regulation and release of non-epinephrine, and of course uptake of non-epinephrine transporter through presynaptic way, the two most important ways where non-epinephrine is removed from the synaptic cleft. Receptor profile wise, you see alpha 2 adrenergic receptors, so stimulation, occupying of alpha 2 receptor in the presynaptic receptor halts the release of non-epinephrine, and that is one of the ways where centrally acting antihypertensive like clonidine are very helpful. By acting alpha 2 receptor presynaptically, they diminish, if you see, the comparatively very little release of non-epinephrine in the neurons. This slide again, similar ones, you see that role of alpha 2 receptors. The non-epinephrine pathways are very similar to, and they actually overlap with the serotonergic pathways. As you can see, it connects the prefrontal cortex. It has basal forebrain area connection. It has definitely connection nucleus accumbens. It connects with the stratum and the cerebellum. And of course the brain neurotransmitter center, or and the spinal cord. It is also associated with a number of important areas, such as the amygdala and the hypothalamus and the hippocampus. So it is connected widely. So by through its wide connection, you would assume that both serotonin and norepinephrine have significant effect on mood regulation. Now, interaction of these two neurons, that's one of the things that we were talking about, that depression is not dictated just by one neurotransmitter. It is the interaction of between these two important neurotransmitters, and we're missing the last one as we talk in a few minutes. And you can see how the neurotransmitters are there. You can see the raphe nucleus. Of course, you have the raphe nucleus and locus coeruleus, the both for serotonergic and for non-epinephrine origin. So non-epinephrine can boost the release, 5-HT release. So via external input from the locus coeruleus, projecting into the raphe nucleus and acting at alpha-1 receptor. This is something important for us to know, both from the examination point of view and from the clinical point of view, is the fact that raphe alpha-1 receptors and non-epinephrine neurons can cause excitatory input from the locus coeruleus projecting into the raphe and acting as alpha-1 receptors on certain bodies. So, and of course, norepinephrine can also reduce the release of via inhibitory input from non-epinephrine terminals. And so it can be both accelerator, as you can see here. If it's stimulating over here, it can accelerate, and if it's on the prefrontal cortex, it can actually cause the breaking up of the neurotransmitter profile. So that's one of the ways that these two neuron systems tend to regulate each other. The next slide is showing about, again, the same regulation of a non-epinephrine and serotonin. Again, you see local coeruleus and you see raphe nucleus, of course, in the brake system, as we just talked about it. We also have the presynaptic and the synaptic regulation here. Close 5-HT accelerator shown here, where non-epinephrine occupies alpha-1 receptors. Now, occupation of alpha-1 receptors actually causes more release of serotonin neurons. You can see it is causing a 5-HT. This is a more kind of schematic diagram. So more 5 alpha-1 receptors that you occupied, the more you release. Okay, more that release non-epinephrine. The other one is similar one, the brake form. You can see that 5-HT brake. Now, if the same alpha-2 receptors are stimulated, it causes the brake. So non-epinephrine has got a significant modulatory effect on serotonin neurons, as you can see over here. Finally, we'll talk about dopamine, the pleasure chemical, as we all know about. Now, dopamine is synthesized from monoamine tyrosine, as you can see over here, and which is transported into the dopamine neurons right here, and a transporter which is very distinct from dopamine transporter known in B over here. So there are two transmitters. One is tyrosine transporter, the other one is dopamine transporter. They are very distinct ones. And again, the rate limiting enzyme, as we the rate limiting enzyme in this scenario is the tyrosine hydroxylase, as we talked about it. And dopamine synthesis is the presynaptic neuron then transported into synaptic cleft, as you can see over here. All these are being in the synaptic cleft, and they are transported towards the synaptic cleft and synaptic from synaptic vesicles, and eventually into the synapse. The similar things, the part of the dopamine is transported. Dopamine transporter is there, the reuptake. The other one is diffusion, and finally into the bloodstream, and finally breakdown by COMT and MAO inhibitors, which are eventually responsible for doing that. Again, you can see that within the presynaptic neurons, they are transported through VMAT2, which is where they are temporarily stored. Very similar blueprint compared to two other major transporters, neurotransmitters. We are talking about a little bit about the neurotransmitters associated with dopamine. Of course, we know that once dopamine is secreted in the synaptic cleft, they can hit either dopamine D1, D2, D3, D4, and of course there is a presynaptic D2 autoreceptors influential in regulating the transmission of neurons, and we have dopamine transporter also, which is responsible for reuptake, as we talked about in the last slide. And the way the D2 receptors, and this is very important in the sense that this is how it is it's regulated. Now, preoccupation of D2 presynaptic autoreceptors causes paucity of a dopamine release. So anytime the D2 receptor is occupied, you see less and less dopamine being transported through the synaptic cleft. The other autoreceptors, D2 dendritic autoreceptor, if you occupy dopamine at the D2 autoreceptors, then again you see paucity of release. So now we have two breaks in dopamine. One is the presynaptic level, the other as the neuron cell body. There are two breaks, as so as to speak, available. Now, in dopamine interaction with serotonin and 5-HT receptor, this is a very interesting chart, a diagram, showing the schematic dependency of dopamine neurons compared to serotonin neurons compared to non-epinephrine neurons. But let's see the interaction between the dopamine neurons and the serotonin neurons. So, for example, now dopamine, as we know, of course, release over here. Now, dopamine neurons are also converging over either through GABA or directly towards 5-HT neurons. Now, 5-HT2A neurons receptors, if you hit 5-HT2A receptors, dopamine break happens. That means inhibits dopamine release. Now, this is again the feedback that 5-HT neurons give towards the dopamine. So, it has got a negative break on dopaminergic neurons. So, stimulation of 5-HT2A receptors will inhibit the dopamine release. Now, 5-HT1A stimulation receptor can accelerate the stimulation of dopamine release. So this is something fairly important for us to remember. So serotonergic, when impinging on dopamine, if you hit the 5-HT2A receptors, it inhibits the dopamine release. And if you hit the 5-HT1A neurons receptors, it'll cause excessive release of that. Now you see also with GABAergic neurons, you have interaction through the GABAergic, and there are other ways they're connected. So a 5-HT neurons leading to a 5-HT1A stimulation leading to excessive release of dopamine, and stimulation of 5-HT2A receptor leading paucity of dopamine release. Now here we are seeing that all three systems are being connected in the schematically. So if this is a prefrontal cortex and these are our two, one is a non-epinephrine neuron, this is a dopaminergic neurons, and in between we have the serotonergic neurons, we do see that there is a connection. There's a brainstem neurotransmitter center connection through GABA, either through locus ceruleus or through a ventral tegmental area. So we have these connections there. Now, if you have GABAergic neurons connecting to dopamine neurons, and we have GABAergic neurons connecting to for non-epinephrine neurons, so you can see the raphe and the locus ceruleus are actually connected, and they influence one over the other. The dopamine pathways are very similar, as you can see here. The dopamine pathways, again, they cover the wide area of prefrontal and frontal cortex, and almost every important area that you can think of, which, if you can see, there's overlapping with serotonergic and non-epinephrine pathways. So this is, in short, is the presentation. I would like you guys to review it, and I'm assuming that you guys have done the pre-test assessment. Reviewing the slides, after reviewing the slides, I would recommend to take the post-assessment test, and then we'll break down into small groups for further discussions about how all this information is clinically correlated. Okay, good luck.